how I would activate my video?
On the left-hand side, I have a button that says stop.
Thank you.
Hello, everyone. Thank you for joining us for this session, which will be a panel discussion following up on our preclinical track. I hope that most of you joining us here were able to catch the presentations, the 5 great presentations that we had on different topics of the preclinical modeling. I did prepare a few themes that we can start off the discussions, but feel free to type in the questions in the Q&A chat panel. Anything that you want to ask the panel, either related to the presentations or the general topics on the modeling in the preclinical stage. Before we get started, a couple of housekeeping items. We take your privacy rights seriously. By attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the Q&A tab on your dashboard at any time. And if you need technical assistance, please use the chat tab. I don't think anybody on our panel needs special introduction. Just to reiterate, we have here our presenters from today: Jeff Woodhead, James Clary, Nader Hamzavi, Maxime Lamardi, Lara Clemens, and a special host, a special guest, Scott Seiler and Liesl Shoda are joining us for this panel discussion as well. So, since we do have experts from different parts of the modeling, from different aspects of the modeling in terms of PBPK, QSP, QST type models, as well as pharmacometrics or more statistical models. Some of you were already presenting some of the challenges, but could you reiterate for our audience, what is the biggest challenge in developing and validating the model type that you typically deal with, whether it is PBPK, QSP, pharmacometric models? I don't have a specific order, so whoever wants to get started.
Well, I can get started if you want, Vera.
Thank you, Max, for volunteering.
Always. I think for my case, and just to reiterate what I've been saying in my presentation, working a lot with locally acting drug products, the main challenge we face, and not just for ophthalmic drug products, is really have access to data to validate the model. Because the model structure by itself, we are trying to mimic the physiology as much as we can. And understanding the physiology, we have access to quite a lot of data on like tissue volume, the tissue organizations, and so on. But then when we try to validate the model by itself and be able to predict the local concentration, knowing the systemic concentration doesn't reflect exactly what's happening locally, this is what the biggest challenge is. We are sometimes kind of limiting in terms of what we can do, although we have ideas of like processes we would like to implement in the model. Not having access to like real data to validate the model is, at least for me in my space, the biggest challenge.
Thank you, Maxime. We have a number of experts from the quantitative systems, pharmacology or toxicology models. Anybody wants to take the floor?
I can jump in. So welcome, everyone. It's nice to have this panel discussion. Thank you for inviting me to participate. So, I think that similarly, we do have, questions about what data sets to use for validation, how to confirm that our models are fit for purpose. But, and there's been actually multiple publications that have sort of looked into different approaches to by which, processes or approaches by which that might be done. But the other part of it, I think that is, maybe also a challenge is the communication with our colleagues that are not in the modeling field. So, helping them to understand, what is included in the model, why it's included, what do we know, what do we not know, how can it be used, and how to interpret the results in a way that is consistent with what the model is designed to do. So I think that, there is, in my experience, there's a lot of appreciation for being transparent in places where, we may not have great data sets. And, there's also sort of those conversations about, how much data do you need? And, if we, there's that sort of balance between you need enough data to do the modeling and to support that and to be able to have that model be useful. But, there still have to be those open questions that the model can then address and support and therefore provide value to the questions and decisions that organization is addressing.
Thank you, Liesl.
So I think from a pharmacometrics perspective, the model we've developed, I think the issue of data continues to be kind of, rings true there too. Some of the mice studies we have for a particular regimen, we only have, 45 mice, which when we're reducing them to one data point apiece and aggregating them together, the more data we have, the more we can really start to see like where those study differences are, what are actual covariates that are driving some of these differences between the studies. Are they just lab differences, or are they actual sources we can quantify? I think too, as well, just there's a challenge in translation from a preclinical model to a human model, depending on the disease severity, the disease state that we're looking at. I know in tuberculosis, we're doing a lot of looking into like how do we correct, where we see these regimens do spectacularly in the mice, and then they get to the clinic and they don't do quite as well as we hoped. Is there some translation that we can do, some modeling to bridge the gap there?
So I'm hearing the theme here. It's the data, and that seems to be the biggest challenge in a lot of these approaches. I think one thing, though, that we need to keep in mind here as well is that even though some of these models do seem complex and data hungry, right, a lot of the data can actually move across the models, right? Some of the information, especially with the mechanistic models, the physiological information, we don't need to keep collecting from scratch every time, right? So as we are building on these models, let's keep remembering that as we are expanding the knowledge, that we don't need to incorporate as much data, but of course, for the compound specific, that is always a challenge.
So this is, let me raise a kind of a tangential, or not tangential, related challenge, which is, we often go into, especially in the world of drug development, with the assumption that all of our animal models are genetically, hence phenotypically identical. In reality, if you actually have the opportunity to look at, individual data points from individual animals, you don't get the same response. And so, if you come in with an assumption that it's like this, that it's going to present a challenge to the modeling. If you kind of take a step back and say, okay, there's going to be some variability, maybe not as much as in humans, but there will be some. And maybe it's the environmental, maybe it's the diet. If you have multiple animals in the same cage, they're going to fight over the food, and one's going to eat a little bit more than the other. Little nuances like that can participate in perhaps overcoming that challenge. But again, just the default setting that they're all the same is, I think, going to for sure present a challenge. And that's not necessarily just for you, James. It's the point you were making. It is across the board. We've seen it as well. If you look at LD50 studies where you give a dose and look at when they die, the animals don't all die at the exact same time, right? They die at days apart. That is the definition of variability.
Anybody else wanted to comment or add to these discussions?
I mean, maybe just one comment in PPK in general is how do we assess variability in PPK? And I think we are still just touching that right now with population simulations and trying to understand what could be the predicted variability in the study. Especially for, I mean, and that's not just for locally acting, but for any PPK model. I agree with you, Scott, like so many parameters can impact either the PK or the PD, because they're linked, like you have some cascade effect. And assessing that variability very early, PPK can help, QSP might be helping as well, population PK for sure, but trying to use modeling and simulation to assess that variability very early, anyone to predict the next stage and maybe exclude some patients or refine the clinical trial or the design in preclinical studies to try to limit as much as possible the variability and really focus on the effect of the drugs and what kind of data we need to obtain to also understand the variability.
Great. That actually, since you mentioned, different types of models, that actually brings us to another topic or the aspects that I expect a lot of people in our audience are interested in. How can these models help to inform each other? Are there any potential for cross communication between these types of models? Can PBPK learn from QSP, population PK, the other way around, and so on? Where do you see the links for these models?
I mean, certainly from the QSP perspective, and PBPK has been a part of the QSP approach, at least ours from the beginning, for at least especially in the QST side of things, where you kind of have to know what the drug is doing and where it's going before you figure out whether it's going to hurt a particular organ or not. So I think there's certainly clearly a lot of information flowing in that direction. I think there's the potential for QSP to kind of feed back onto the PBPK, and as the physiological biological changes in modeled in a QSP model are undertaken, and that comes back onto the PBPK and helps you learn a little bit something about what's going on PBPK wise. So certainly in that, for those two models, they're very intricately linked.
Yeah, and I think PK, PD models, just looking at like compartmental models, we can gain a lot of information, particularly from PBPK, and particularly when parameter uncertainty is really high, and we know that there's forces driving those particular parameters, and looking back at PBPK models that have been utilized, can give us some good estimate to ground some of these estimations in. So I think we've seen even in the recent few projects I've been working on, kind of blending some aspects of PBPK into population modeling as the model becomes more complex to describe the data more accurately.
So I actually have a follow-up question on that one. When you talk about blending the PBPK into PopPK models, do you actually look at trying to build a PBPK model, look at the critical mechanisms to help you drive the structure of the PopPK model, or is it more of a general type of information about the mechanisms?
I think that opportunity definitely exists as these models become more and more complex, and the model more accurately. But at least for the projects I've been working on, is more of a general just driving as we have complex systems like maybe an eye, or in particular is what I'm thinking of, something like that, where it kind of becomes necessary to look into the literature and refine those values, because just with the data available, you're probably not going to get good estimates.
Okay, thank you.
If I can rebound on that, I think also PPK, we could also be helped quite a lot by population PK to somewhat rank the study we use in model development, because we tend to use a lot of study to either validate or develop or verify the models. And sometimes there is some study where we are pretty far off the data and we don't have the luxury to put an inter-study viability. But trying to use the population PK mindset to compare those studies and maybe try to look into, is there something very specific to a study that is not described in the publication or is not described in the data, and really bring that study apart from all of the others. I think that could help us to both ways, for us to develop the model and make sure we use the best studies, and also when we go to the authorities, explain why we didn't use a certain study or why we kind of give a certain power to each studies, because this one seems to be an outlier when we compare it to everything else. And right now, we tend to go with yeah, this one is weird in a way, but I think the pop PK concept could be applied and give us like a statistic reason to say, okay, this study seems to be an outlier. We still try to incorporate it. It didn't work. Don't judge us on this one. Judge us on the rest that makes sense.
Yeah, that's a very good point. Yeah.
I want to get in on the fun too, since we're all, having giving such thanks to pop PK modeling and their methodologies. So something that we borrowed from the pop PK world on the QST side, specifically with DILIsym, is there are a number of situations where a drug has been given and you'll see rises in elevations in liver signals, i.e. indications of liver damage. But one thing that's difficult to quantify in humans is the magnitude of the damage. The biomarkers are elevated, but how many hepatocytes have been lost? And so we actually, again, really borrowing largely from the pop PK world, basically looked at the biomarker profiles, kind of went back. We knew the kinetics of biomarker release is a consequence, a function of hepatocyte loss, the rates of necrosis, for instance. And then we were able to ultimately infer predict, i.e. the magnitude of hepatocyte loss over that period of time based on that dynamic, et cetera. And then, so you can do that on an individual by individual basis and, i.e. helping out those specific patients, but also kind of on a population level, right? And so really understanding the magnitude change within it. It's, I think, a really nice example, again, of this cross-fertilization that about which, you've asked.
Yeah, I'm glad to hear that everybody is utilizing all of the data and different types of models, right? And that's what it all comes down to. Make sure that you utilize all the information and sort of totality of the evidence about your compound in order to make the best decisions. Anybody else would like to chime in on this discussion topic before we move on to the next, or start addressing some of the additional specific questions to the presentations?
Yeah, just one thing I wanted to mention was about the crosstalk also between these platforms. So we started with PBPK modeling to estimate the exposure, and then we do our QST modeling, for example, in line with my poster presentation, so we know how much of cell loss we get, and then we are thinking about like how integrating that cell loss back to the PBPK model and readjusting the exposure. So in a way, the crosstalk between these different platforms is also helpful to have a dynamics in terms of the cell numbers and the functionality and the exposure.
Yeah, that is a very good point. Yeah.
It actually works in the opposite direction as well. If you have treatment that helps relieve the cell loss, the pressure of the cell loss, then you have, in theory, more cells capable of clearance and metabolism, right? So it isn't unidirectional, it's bidirectional, potentially.
Sure. Yeah.
Okay, so follow up on this, actually, when we talked about what is possible to do with these models, right? Can some of you talk a little bit about where these models are currently utilized heavily, especially in the preclinical stage, but if you want to touch on the other stages as well, and especially where do you think that they are underutilized right now, where you would like to see these approaches to be utilized more to improve the drug development process even more, and how do we get there? How do we promote the utilization of these models within the industry? Scott, do you want to give it a shot to get everybody started?
Yeah, go for the easy one, right? The person you know will talk. There's actually a really interesting. So thus far, I don't think we've really brought up QSP modeling, i.e., focus on efficacy. One of the biggest challenges in many situations, different therapeutic areas, et cetera, are that the animal models, not mathematical models, the animal models of the disease state are far from representative of the human patients of that disease state. There are species differences. There are just differences in developing, kind of encouraging the animal to develop that disease. And hence, the response to treatment that you see may be different in that animal model versus the human model. So it's really very interesting then to consider and compare what about an animal, comparing an animal model versus a mathematical model of the human disease, i.e. a QSP model. In theory, you will be able to make a prediction that is perhaps more relevant to the human using a validated mathematical human model, right? Now, there's some, a multitude of questions that can arise. And I think perhaps the ideal scenario is to have one of each. If there is an accepted animal model that is really well established and used across the board, you develop a mechanistic model of that, as well as a mechanistic model of the human, and then you're able to do simulations in both. One example of at least the first half of that is a paper published, work led by Dr. Liesl Schoda with the NOD mouse for type 1 diabetes. Again, it was a predominant model. If you move into other areas, there are 17 different animal models. And so it becomes more difficult to do something like that. But if there's one on which everyone agrees, one animal model, develop a mathematical model of that, as well as the human, and you're able to facilitate that translation better.
Another place where I see PPK could be used in a preclinical development is actually before conducting any studies, because the price of animals is increasing, and there is a global concern now to not kill as many animals as we did in the past. And because PPK really have this bottom-up approach, it is really an opportunity to predict in advance what could be the expected concentration, and also predict the number of animals we need to answer the question we are looking for. So for example, for formulation optimization, it would be possible to just design one preclinical study, use that to verify the model, and then conduct X numbers of virtual studies to define what would be the target formulation we need to achieve and to obtain. And then once we set out on that, maybe run another preclinical study to validate our model, just to make sure, and before going to first in human and so on. But using PPK model can really help to reduce both the cost of preclinical development and also the cost in animal, which is becoming a global ethical concern.
So there's actually looks like Brett made a comment in the chat about the clinical budgets and just not having the money to spend on some of the larger modeling projects really early in clinical development. And Brett and I were actually having a discussion about this with some of our collaborators yesterday. And I think there's a lot of value in kind of designing modeling around what is already going to be available to those teams in very early development, whether it's just machine learning and structure, base things. The ADMET predictor is already kind of in that space, and I know we're trying to move that direction ourselves, whether it's in some of the omics data or whether it's simply in demonstrating the value of conducting sensitivity analyses and saying, hey, here's what you need to look out for with these compounds later on in clinical development. And if you get to point X and see something, then you can stop things early before you see that means you'd see something in the clinic. So any of those particular approaches could adapt something that's been primarily a clinical tool up to this point into that early preclinical space.
In terms of applicability of these models, we actually have here a specific question for Nader about the possible applicability on the kidney-induced kidney injury approach on other nephrotoxic drugs. And if you've explored some of the other drugs, how much more you might have done on that?
Yeah, thanks for the question. So I think we have already included like 13 different compounds in Renosym V1A, including cisplatin. So the list includes gentamicin or acyclovir or negative controls as well, such as acetaminophen. So we are trying to include those known nephrotoxic drugs that are causing or showing signals for different toxicity mechanisms, so we can test the model pretty well. Yeah, and I think we as the model is also progressing, and for future versions, we may include more compounds if there is interest.
Right, thank you. Going back to the applicability of the models, I actually also wanted to ask Clara, since you did a presentation on the large molecules, right, on the biologics. What is, in your view, the application in the biologics area, and where do you think it is heading? What would be the next stage of applications of these models, especially for biologics for the large molecules?
Yeah, I mean, I think this first phase has been focused on proof of concept and working with molecules that are kind of already out there and showing that we can take these models and reproduce their hepatotoxic potential. But naturally, the place that we'd go after that is molecules that are still in development and trying to take molecules that we maybe don't know how hepatotoxic they might be, and working based on what we know about their targets and what their downstream target effects are, try and get a sense of, do we expect that this will be damaging to, in this case specifically, the liver, which I think is a non-trivial question. And there's a lot of moving parts depending on, particularly there's a lot of investigations into all sorts of monoclonal antibodies, and they can have all sorts of fun effects. And so that's going to be a really valuable direction for this to go, and has a multitude of applications.
Thank you.
If I might just go back to going back to the small molecule that we were discussing previously and some of its additional applications. We talk a lot about how, as modelers, we always wish that there was more data. But I think one of the nice things about modeling is that it gives us an opportunity to really explore the parameter space and get a sense of then what data do we actually need. So, we talk a lot about what data do we have that we can use, and then what do our models tell us about data that we maybe couldn't obtain. But we should also think a lot about, using these to inform what data would be most particularly helpful, so we can focus our efforts on gathering the data that would most validate or most support particular mechanisms, and then also get a sense of, okay, we can do these sensitivity analyses and get a sense of maybe what pieces are not going to be as critical in determining whether a particular molecule is hepatotoxic. We have many different mechanisms, and they all have different components. Do we actually need data to tie down particular pathways, or is variability in that particular pathway maybe less important? And I think that we have a really good opportunity with these simulated populations and all of these different platforms to answer those questions and really inform some of those basic connections.
So build on that. Yeah, so one of the points that came up, I think, when you asked the question was how has it been used, in the preclinical space? And I would say that, it's varied quite a bit. There have been several occasions where we've taken a QSP model to investigate the potential, therapeutic potential of a particular target with very minimal data sets available, or sometimes no internal data sets available, and really just relying on the publicly available literature. So it's, sort of more of a focused sensitivity analysis. But, there have been projects where you can take, 10 targets and come up with a ranking. And because maybe a smaller group, but because you're doing some things in populations that have variability, you might be able to do some secondary analysis to identify, what are the key characteristics of the responsive patients, for a particular strategy. And I think that, being able to provide that type of, insight is something that we've certainly done. Often those are a little bit harder to talk about publicly, and as well as, put out in the form of publications. But certainly I think the possibility of helping to, bringing additional tools to be able to prioritize some of those efforts, while drugs are still in preclinical, is certainly something that we could, we have done, but we could do more of.
Yeah, and I think building off that, I mean, that's kind of what the heart of my presentation was trying to get to is we have a lot of data, and we know how these trials are operated, and how can we refine them to be more cognizant of our resources, and then also help inform these no-go decisions for these different regimens and that extends, I think, in our case it's kind of nice. TB has a lot of data involved in it, and these trials are still ongoing, so we can utilize these mice models. That's sometimes harder when it's a new clinical compound, but I think there is room kind of in the same space of like the PBPK. If there is data for disease states or known mechanisms, then there's value in helping to design these trials to maximize the information while still being a good steward of the resources that are available.
Vieira, can I ask a question of the group? Do you mind?
Sure. No, go ahead. This is a discussion. You can ask questions each other.
I didn't, you're the moderator. One thing that comes up obviously with preclinical more so than clinical, although a little bit there as well, is the use of gene chips and tons of expression type data. And how do you in this preclinical setting? This is a vast. We just said maybe we're data limited, and then the folks collecting the data say, "My goodness, we have so much data." Is there what sort of approaches kind of in each of these disciplines are we using to help kind of bridge that? The types of mRNA relative expression sort of data sets, and how are we applying each of our modeling disciplines to making use of those and within the context of fluxes and mathematical quantitative relationships. So if I may, I'll ask Max to answer first.
Well, in BBK model, so we have specific physiologies for each animals. all of the data we gather through multiple projects, we will incorporate them in the studies. So when you talk about mRNA, for example, if we have information about expression level of enzymes or transporter in specific tissue of an animal, the idea is to integrate in the model so we can predict then the exposure or the clearance, whatever it is referring to. I would say as we gain more knowledge and as we get access to those data and we implement them in the model, what it could help us is to go back to your initial point regarding variability, is to better understand the inter-animal variability and trying to get that out of the equations. So when we want to predict what could be in human, because the enzyme expression level may not be the same between animals and human, like we could maybe be able to better predict the variability in human because we understand where the variability is coming from in animals.
Now I'm going to flip the table or turn the table. Vieira, do you have anything to add to that?
Yeah, I was just gonna say that, to add to that, I don't think that we've ever been in a situation where we had too much data, right? We might have been in the situation where we are thinking that we have too much data if the data appears to be conflicting. That is something that we deal with quite a bit, but it goes back to the variability. As especially when the data just starts coming up and we appear to have only few data points and they seem to be conflicting, not in line with each other. But as we start filling in more information and more information is coming out from publication and so on, it starts filling in the gaps, and then we are able to figure out why those initial points did not seem to agree with each other. And so, of course, these models are ever evolving. Right? We start with whatever information we have. It's very little initially, and keep improving them as some new information comes up from various research labs and so on. But yeah, I don't think we've ever reached the point that we have too much stuff.
Now, when you have an abundance of data, isn't that just ripe for something like PK/PD modeling? So, James, have you ever had, you and your colleagues in Buffalo and elsewhere, have you much experience kind of relating those gene expression types data to exposure?
I think it definitely depends on the compound and the drug in particular, but there has definitely been some linking of covariate values to different gene expressions, usually more of a poor metabolizer, more of a categorical type, poor metabolizer, extensive metabolizer. But I have seen some publications where that has become something of a continuous covariate measuring different enzyme values. I think the big thing that comes to mind is just for us particularly is having an open and honest conversation with whoever is providing the funding for the model that we're making, and asking them what data do you have, are there mechanisms that you think would have some impact that maybe we're not thinking about because we're thinking about this more on a population scale, and can we build this mechanism in because you have that data that we didn't really know about before. So I think it's really kind of going back to I think a bit what Liesel said, having those conversations with the team to really understand what's available and how that might help you quantify some of that variability.
Mm-hmm. So, one of the advantages of these types of data are they're, kind of fundamentally they are mechanistic. So I turn to one of our mechanistic modelers, Jeff. How have you in the past handled kind of the classic gene expression data within the context of fluxes and variety, transporters, et cetera? Is there a way to make use of data?
There is, from a qualitative perspective. I think it's really difficult to quantitate that, right? There's if there's protein levels, that makes it a little bit easier. But in terms of just kind of determining where your outcomes are, where your outputs are, matching things to outputs, and then you can kind of use gene expression as a way of kind of bounding your variability. That if there's 3 or 4-fold difference in gene expression within a population, that when you're building your population model, you can build in that general range of variability. That's how we've used it in the past. I'm sure there's more quantitative ways to relationships that can be backed out if you have enough data, or if you have enough inputs to relate to outputs, and enough gene expression data, and in there you can generally you can pull something together. But just kind of in isolation, it's very difficult to make the mRNA levels to protein to activity relationships purely quantitative in any really reliable fashion without knowing where you're going or where you're coming from.
Let's see, there's transcriptional regulation, there's post-transcriptional regulation.
Right.
Post-translational, there are pools, sequestered. It is, varying degrees of complexity, and it would depend then on specifically the gene product and the gene and kind of that path, right? Each has their own unique one. But that doesn't mean that it's not doable, right? That's
right.
It's there's still value in those data, right?
Mm-hmm.
So to build on that, and going back to the importance of having conversations with, your collaborators, your colleagues, your clients on the other side, sometimes this type of data, right, tends to be quite mechanistic, and it can get down to a level of detail that sometimes we are not representing in the models. But, depending on the interests of the group, that little bit of information might be just the bit of leverage that they need to be able to pursue a hypothesis of interest. So for example, in the world of lung fibrosis, there is this phenomena that's been characterized called epithelial to mesenchymal transition. I think I've got that right. And it's very interesting. It is a potential reason why the lungs become more fibrotic, but quantitatively to understand, what is the contribution in people, how much does that happen? How fast does it happen? Does it vary based on where you are in the disease stage? Those are good questions, tough questions, and most of the data are not available to address it. So you might not include EMT as sort of a standard contributor because you don't know how much it contributes. But there have been, there's an increasing body of literature available to look at things like expression or fluxes or what have you, or you're looking at, a particular gene that is more indicative of this phenomena. And maybe that could provide, the impetus to explore that particular hypothesis. And if the data are quantitative, it might even give you some guidelines, as Jeff had alluded to say, okay, we're going to explore this and we're going to allow this to account for, 10% of the population, or, between 1 and 10%, or what have you. So I think there are definitely opportunities that are sort of opportunistic based on the interests of, the project team, as well as, sort of the availability of data.
If I can jump in with another question for everybody is, you've all have been doing this for, some of you shorter period of time, others, for a number of years now, and working with the collaborators, helping them to address these questions. Have you seen the shift in the attitude or in the approach that they are taking to the modeling over the time that you've been using or doing the modeling, helping them to address different types of questions in the drug development process?
Oh my goodness, massive shift. I'm probably guilty of telling this story too many times, but the tale of QSP, QST is almost exactly the tale of pharmacometrics and PBPK, but each one is time shifted, where you're introducing a whole new technology to a world of laboratory experts who, what do you, math? Yes, no, kinda. And so there's this slow adoption. And I will share my experience in QSP in 1999, before pretty much anybody was using it, it's certainly not broadly. This is what we do, was met with, you can't do that. Right? And 2 years later, you can do that. 2 years later, oh, we could use that. And then, slowly the adoption curve, you're climbing up that, right? And again, each one, I think each discipline of modeling, even though they've been established for decades prior, to really incorporate it within kind of the true process and to have everybody understand its exposure, as in educational exposure, familiarity, it's, success breeds confidence, all those things. And I think we've established across many modeling disciplines a lot of those success cases that allow that confidence, hence the adoption to really have occurred. And I'm not exaggerating, I literally heard, you can't do that, more than once.
Yeah, we've seen that in the mechanism. Well, I haven't seen it because I haven't been here at the beginning of the Gastro Plus, but from what I've been hearing at the beginning, it was, yeah, you can't do that, right? But over time, now it is one of the tools used for the predictions. So yeah. We are getting close to the end of our session. Thank you all for your participation and for your valuable insights and comments. Anybody has anything as the final words of wisdom for our audience before we close this session?
Maybe just one thing. It's a comment made by someone at the FDA back at their meeting in September. If you tried everything and you do not have the answer and you have not tried modeling yet, you should try modeling, because the answer might be here. So if the FDA says it, I mean, it's not us.
Hopefully pretty soon we'll get to turn that around. Try the modeling first. Don't try everything else. Do the modeling first, and then see if you need to do anything else.
All right. But I think it goes back to one of your points, is how we reach out and educate people about what the model can do. So we don't get the answer you and Scott just referred, but actually people are more aware of what the tools can do. Right. And those workshops, more presentation, more publication, is part of the answer.
Excellent finish to this session. Thank you again all for your participation. And thanks to everyone for listening in and submitting the questions on this discussion. This session has been recorded, and I think it will be also available for playback on our website and YouTube channel. This concludes the session, but the day is not over. There are still additional sessions going on and booths that you could visit. So feel free to go and attend the remaining sessions for today. And I'm sure we'll see you in a full force tomorrow again. Thank you, everyone.
Thank you.
Thank you.
Thank you. Bye-bye.
Thank you.
Bye.
Bye.